Good evening, people. Can you hear me clearly? Can you hear me? Yes or no? Please send a message. How are you? Good evening, everyone. Komuda. Komuda. Am I audible? Clearly? Right. So we have to discuss today the paper number 10. Paper 10. Right. Paper here. 8 can open to the paper 8 and 10. Right, people, I gave you paper 8 marking scheme last time. Last Saturday. 10. Right, people will discuss multiple choice questions first. We discuss the theory part of this theory last time. Right, people, this is the paper. So we'll right, first question. First carbon dioxide acceptane C4 plants. First carbon dioxide acceptane C4 plants. First carbon dioxide acceptane C4 plants. We know, people, that is the PEP, phosphoenol pyruvate. We can take the answer as the third one. Question number 2, which of the following statement is correct regarding photosynthesis of C4 plants? Photosynthesis of C4 plants. They are asking correct statement regarding photosynthesis of C4 plants. Right? Only malate transported through plasmodesmata. Only malate transported through plasmodesmata. Following is correct regarding photosynthesis of C4 plants. Only malate is transported through plasmodesmata. Malate transported through plasmodesmata. No, pyruvate also need. Pyruvate move to, right? Pyruvate move to. So not only malate, and PEP carboxylase is, just give me a second, people. Right. PEP carboxylase is involving in carbon dioxide, only malate, wrong kilagamu. PEP carboxylase is involving in carbon fixation at the bundle sheath cells. PEP carboxylase bundle sheath wrong, the mesophyll cells, wrong statement. Decarboxylating enzymes, Decarboxylating enzyme function in the bundle sheath cells. Decarboxylating enzyme function in the bundle sheath cells. Bundle sheath cells, we can see that people malate is converted back into pyruvate. So some enzyme need to be there, and that releases carbon dioxide. That is correct. Rubisco operates exclusively at the mesophyll cell. Wrong, the bundle sheath cell. There are no photorespiration in C4 plants due to absence of Rubisco. There are no photorespiration in C4 plants due to absence of Rubisco. No photorespiration in C4 plants due to absence of Rubisco. Now we learned that photorespiration present, but it's a minimum, need. So best answer, people, for the second question, third one. Question number 3. Question number 3. Correct statement regarding C4 pathway of photosynthesis. Correct statement regarding C4 pathway of photosynthesis. Initially, carbon dioxide is fixed by PEP in the bundle sheath cell. Carbon dioxide is fixed by PEP in the bundle sheath cells. Correct statement regarding C4 pathway, need carbon dioxide fixed by PEP in bundle sheath cells. Wrong, need mesophyll cells. It was a fixed carbon fixed carbon is reached to bundle sheath cells as malate, which are more stable state. Okay, and then Calvin cycle take place in mesophyll cells of this pathway. Calvin cycle take place in mesophyll cell. Wrong, the bundle sheath cells. Generation of PEP passive process. PEP is a passive process. Regeneration need energy, the wrong. And then primary product of this pathway is 4-carbon carbohydrate oxaloacetate. The primary product of this pathway, 4-carbon oxaloacetate. Primary product of this pathway is 4-carbon carbohydrate. Wrong, oxaloacetate correct, but it's not a carbohydrate, it's just an acid. So wrong statement. So we'll take people second one as the answer. Second one as the answer. Question number 4. An adaptation developed during the plant evolution. An adaptation developed during plant evolution by C4 plants is reduced photorespiration. An adaptation developed during plant evolution by C4 plants to reduce photorespiration is that they are bundle sheath cells. An adaptation developed during the plant evolution by C4 plants to reduce photorespiration is that they are bundle sheath cells. Adaptation developed during plant evolution by C4 plants to reduce photorespiration. Fix carbon dioxide twice. Reduce photorespiration. To fix carbon dioxide twice are relatively smaller in size. Photolyse water molecules. Bear chloroplast rich in grana. Reduce amount of photosystem. Reduce the amount of photosystem. Photosystem 2. Yeah, photosystem 2 is better, neither. But reduce the amount of photosystem. Because photosystem is involving in making of photosystem involves in splitting of water and then produce people oxygen, neither. So answer is 5. Answer is 5. Question number 5. Which of the following statement regarding photosynthesis is correct? Which of the following statement regarding photosynthesis is correct? Rubisco enzyme is effective only in C3 plants. Rubisco enzyme is effective only in C3 plants. Which of the following statement is correct, Kilahanne? Rubisco enzyme is effective only in C3. No, it's like more effective C4, neither. Why? Because the photorespiration occurs in C3 plants. PEP carboxylase is present in both C3 and C4 plants. PEP carboxylase is present in both C3 and C4 plants. No, that's wrong. In C4 plants have expanded physiological function. In C4 plants have expanded physiological function, that is correct. In C4 plants, carbon dioxide is fixed in the cytosol of the bundle sheath cells, mesophyll cell, wrong. Enzymes are involving in decarboxylation step in Calvin cycle. Enzymes are involving in decarboxylation step in Calvin cycle. Enzymes are involved in decarboxylation step in Calvin cycle. Calvin cycle decarboxylation wrong, neither. Carboxylation, neither. Calvin cycle decarboxylation does not occur. In what you call Krebs cycle, decarboxylation occur, not in Calvin cycle. Ne? So we'll take it as wrong statement. So we got the answer here. Third one is the answer. Third one is the answer. Question number 6. Which of the following statement is false regarding kingdom fungi? False regarding kingdom fungi. All fungi are multicellular except yeast. All fungi are multicellular except yeast. Wrong, neither. Why? Chytridiomycota we have unicellular ones. So wrong statement. Different species live as decomposers, parasites or mutualistic. Different species live as decomposers, parasite or mutualistic. Correct. Multicellular fungi produce mycelia, form fungal hyphae. Chytridium is also lives in aquatic fungi. Also lives as an aquatic fungi. Rhizopus mycelium possess aseptate hyphae and produce sexual spores endogenously. Rhizopus mycelium possess aseptate hyphae and produce asexual spores endogenously. Correct, neither. Rhizopus. So we have the answer now, people. For the 6th question, first one is the answer. Question number 7. Question number 7. Which of the following statement regarding to kingdom fungi is correct? Sporangiophores are endogenous and genetically diverse. Sporangiophores are endogenous and genetically diverse. Sporangiophores are endogenous. Sporangiophores are endogenous and genetically diverse. Sporangiophores are endogenous. Sporangiophores are endogenous and genetically diverse. We said people sporangiophores. Sporangiophores we can take in endogenous. Which statement regarding is correct, Kilahanni? Sporangiophores are produced in ascomycota, right? Endogenous neve then exogenous neither. Wrong. Question number 2. Statement 2. Zygosporangium produce genetically diverse endogenous spores. Zygosporangium produce genetically diverse, genetically identical, wrong, genetically diverse, because it's a sexual reproduction process. And then people, 8 ascospores are formed within ascocarp. 8 ascospores are produced within ascocarp. Ascocarp, 8 ascospores are produced inside ascus kyonang hari, not within the ascocarp. Inside the ascus neme. Ascus is produced inside the ascocarp. Wrong, neither. Basidiocarp has basidia. Basidiospores are genetically diverse. That is correct. Conidia are produced in sexual reproduction. No, asexual reproduction. So we have the answer. 7th one, 4. Question number 8. Question number 8. What is characteristic that differentiate agaricus from ascomycota? What is a characteristic that differentiate agaricus from ascomycota? Agaricus basidiomycota neda. Ascomycota and basidiomycota. Production of exogenous asexual spores. Ascomycota produce asexual exogenous. Agaricus do not produce asexual spores. So that is therefore wrong. Having dikaryotic fungal mycelium. Having dikaryotic fungal mycelium. Who's having dikaryotic fungal mycelium? Agaricus has the dikaryotic. Ascomycota not common, neither. Wrong statement. Production of 8 basidiospores on basidium. Production of 8 basidiospores on a basidium. Basidium is producing only 4, wrong, ne? Production of sexually differentiated gametangia. Sexually differentiated gametangia differentiate agaricus from ascomycota. Ascomycota has sexually differentiated gametangia, that is correct. Right? Sexually differentiated gametangia. But you don't see any gametangia in agaricus. No gametangia in agaricus. That is also wrong. So we don't have a proper answer for this. We don't have proper answer for this. But then we can take people, the second one, having dikaryotic fungal mycelium. Right? Agaricus has a dikaryotic mycelium, correct. Ascomycota is not very clear. Present in a short time, but it's not very clear. So we'll go for the second one as the answer. Question number 9. Question number 9. Formation of exogenous asexual spores. Multicellular xenocytic. Dikaryotic mycelium is dominant. Septae found only in where reproductive cells are formed. Septae found only where reproductive cells are formed. Phylum for above description. Now, formation of exogenous asexual කියලා කියන්නේ asco නේද? Multicellular xenocytic කියලා කියන්නේ zygo. Zygo. Dikaryotic mycelium dominant කියන්නේ basidio. Septae found only where reproductive is zygo. Right? එහෙනම් this one has to be නේද? Multicellular xenocytic will take people for chytridiomycota නේද? We'll take that for chytridiomycota. We'll take that chytridio. So therefore, first one must be asco, second is chytridio, third is basidio. So the answer is third. Question number 10. Select the correct responses regarding plant growth regulators and its function given below. Select the correct response regarding plant growth regulators and its function given below. Auxin stimulate pollen tube growth. Auxin's pollen tube growth wrong. That is by gibberellin. Auxin retard leaf abscission. That is correct. Ethylene promote desiccation tolerance. Ethylene promote desiccation tolerance wrong. Neither. Abscisic acid. Neither. Abscisic acid stimulates seed germination. No. Inhibit seed germination. Cytokinin inhibit growth. No, it's growth promoter neither. So I think 10th question we can go for the second one as the answer. Question number 11. Which of the following plant growth regulator stimulate stem elongation at low concentration and promote formation of the root? Which of the following plant growth regulator stimulates stem elongation at low concentration and promote formation of root? And promote formation of root. Following plant growth regulators stimulate stem elongation at low concentration and promote formation of root. Stimulate stem elongation at low concentration. Stimulate stem elongation at low concentration and promote formation of root. Right, we didn't discuss this in our theory part. Auxin, we mentioned is affecting on the root formation. Auxin, it's mentioned affecting in root growth in low concentration. All right. Auxin promote auxin. Auxin retard stimulate stem elongation in low concentration. Formation of lateral and adventitious roots කියලා තියෙනවා. Auxin වලට following plant growth regulates stimulate stem elongation at low concentration. Stem නේ අහන්නේ. එහෙනම් auxin නේද? Stem elongation auxin. Question number 12, people. Which of the following statement regarding plant responses to internal and external stimuli? Correct. Which of the following statement regarding plant responses to internal and external stimuli? Correct. Cryptochrome are red light photoreceptors. Wrong, neither. Cryptochrome blue light, neither lamai. And increase of far red to red inhibit branching. Increase of far red to red ratio inhibit branching. Inhibit branching. Increase of far red to red ratio inhibit branching. Now this resource book point is wrong for that, but anyway we have to go accordingly. This says direct sunlight increases the proportion of far red to red. Increase of far red to red inhibit branching. Exposure to direct sunlight increases the proportion of far red to red and thereby stimulate branching. Increasing of far red to red inhibit never stimulate kilagannone. But the resource book is wrong. But anyway, we can't do anything now. We have to go accordingly. Right, so we'll take it as it is given. Plant detect gravity by settling statolith. That is correct. And then statolith are found in cell elongation zone. Never wrong in the root cap. Red light photoreceptors are reasons for slowing down of hypocortical elongation. That's blue light, not red light. So we have the answer now, people. Third one is the answer. For question number 12, answer is third. Right? Question number 13. Question number 13. Some statements regarding stress response of the plant are given below. Correct order of drought, cold, salt kilahani. Leaves roll kilaken drought neither. Drought. Development of salt glands, salt. Increasing of ratio of unsaturated fatty acid, cold. Production of toxic compounds, biotic. So, drought A. ඊට පස්සේ cold නම් ළමයි C. ඊට පස්සේ salt නම් B. D නේ. DC ACBD ACBD second. Question number 14. Which of the following is correct regarding stressors in plants? Stressors in plants. Which of the following is correct regarding stressors in plants and their responses? Drought stress increases the synthesis and release of gibberellic acid. Drought stress release increase synthesis and release of gibberellic acid. Neither. Abscisic acid. Neither. Wrong. Cold stress increase the proportion of unsaturated fatty acid of the plasma membrane. Cold stress increase proportion of unsaturated fatty acids. Unsaturated, correct. Production of solutes that can tolerate the low concentration. High concentration, wrong. Having root hair, tricks and trichomes, biotic stresses. Root hairs, trichomes, ne? And then salt glands keep higher water potential in the cell than the soil solution. Keep higher water potential in the cell. Salt stress. Lesser water potential, wrong. So we got the answers. 14th, second one is the answer. Question number 15. Pair of plant growth substances that include seed germination. Seed germination, people. Gibberellin and cytokinin. Fourth one is the answer. Gibberellin and cytokinin. So that's the structured assay part. That is structured assay part. Right. People, we have to now move to the, sorry, that's the MCQ part. We have to now move to the structured assay part. Right. How are you doing? Do you have term test? Any term test, people? Any term test? No? No term test, ne? Yeah, please chat with me. Do you have term test? No? Yes, when? Fatima, call me when? You said this month? End of this month, ne? End of this month. Okay. Right, now let's go to the structured assay part, people. Now you have to tell me the next three sections that we should take for discussion. Next three sections, but we couldn't discuss. Ritika says plant nutrition, vertebrate enzymes and plant nutrition. Life cycle of plants. Fatima Faumi says life cycle plants. Any other suggestions, people? Any other such? I think we have to make it little narrow, so that we can finish discussing. Because if you take plant life cycles, I think Fatima, it's going to take the whole day, just for discussion of 5 life cycles. Little long, neither. We have to take that plant life cycles. If you're taking plant life cycles, then we can go only with plant life cycles. That's too boring, we'll divide the plant life cycle. Maybe we'll take Neprolepis and Pogonatum, right? 2 plant life cycles and then enzyme and maybe vertebrates, like that. Otherwise, it's going to be too much. Right? Plants, photosynthesis, I'll take some other time. We'll come back for photosynthesis in another cycle and we'll have a discussion. I think we'll go with enzymes. We can go with Neprolepis, Pogonatum, So can't we include plant nutrition? Plant nutrition was the big problem with that. Plant nutrition, mineral nutrition, those things, neither. We'll take Pogonatum, Neprolepis, I need enzyme vertebrates. Vertebrates, is it vertebrates? Okay, we'll take vertebrates. We'll go like that. Enzymes, Pogonatum and Neprolepis and vertebrates. Is that okay? Is that okay? Plant, yeah, we'll go like that. Right? Plant nutrition will take in the another round. Right? We'll take plant nutrition in another round. All right. The lot of people are so silent and no support at all. Right. You have to support this process. True, it's a little tough, neither. Right. True, it's little tough. And that's anyway, people, how A level is, we have to participate in this work. Right. Somehow we have to now today less attendance also. We'll take care of that on Saturday, and we'll see. We have to put now a lot of pressure on them to make sure that they join with the discussions and all. Right now, a lot of students are there. They don't want to discuss questions at all. They straight away people they want just the theories. That's all. Right? That's not going to work. You need to do questions and the studying everything together, right? So that's a very wrong strategy. We have to right, promote this. I will go to the paper discussion structured essay part. Structured essay part. Structured essay part. Following diagram shows photosynthesis. Name A to G. Name A to G. What is A, people? A. Where is A? This one. We'll enlarge it a little more. So in plant life cycles, do we only have to study Pogonatum and Epilobi? That's correct. Only those two, right? Then we'll take that to another paper. Otherwise, I won't be able to discuss that with you all in the next one. We'll take consecutively the plant nutrition, plant life cycle continuous consecutively. So the coming paper, paper 11, and also for paper 12, right? So that we can finish it in a month time, right? So let's discuss this. A, what is A, people? A is oxygen, neither. A oxygen. A oxygen. A oxygen. B water. C, this one, neither. C, P680. D, primary electron acceptor, D. And E, ATP. And F, P700, this one. And G, NADPH, right? NADPH. Hope you have got that part correct. What happens to carbon dioxide in presence of hydrogen? What happened to carbon dioxide in presence of hydrogen of water during photosynthesis? What happened to carbon dioxide in presence of hydrogen of water in during photosynthesis? Now, in the resource book, people, they have mentioned in presence of hydrogen, carbon dioxide reduced to simple sugars and then made using energy. So this is what they say: reduce and simple sugars are made. That is your that should be your answer. Which visible lights are absorbed by chlorophyll A? Which visible lights are absorbed by chlorophyll A? Blue and red lights. What is meant by absorption spectrum? Graph of the relative amount of light absorbed at different wavelengths. Next one, what are photosystems? What are photosystems? Complexes formed by organization of chlorophyll, other organic molecules and proteins in the thylakoid membrane. Right? So here marks are given: complexes formed by organization of chlorophylls, these complexes, other organic molecules, and proteins, right? What are two complexes found in photosystems? Reaction center complex, light harvesting complex. 2 marks for that. In which photosystems the splitting of the water take place? Photosystem 2. 1 mark for that. What is the significance of splitting of water in the photosystem? You mentioned above, right? Significant splitting of water to yield electrons. Indicate the molecules that form during some photo-excited electrons from photosystem 1 use as an alternative cyclic pathway. I indicate the molecule that forms during some photo-excited electrons. Again, what is formed during the cyclic electron pathway? ATP. Give 3 significances of C4 pathway over C3 pathway. C4 pathway over C3 pathway. C4 pathway improves the efficiency of carbon dioxide fixation at low carbon dioxide concentration. Then exhibit the water use efficiently. Ilangada leads to better nitrogen use efficiently. Question B. What is the cell wall material present in fungi? Chitin. What is absorptive nutrition seen in fungi? What is absorptive nutrition seen in fungi? So you describe the absorptive nutrition here. Secrete extracellular enzymes which aid in breaking down of complex molecules into smaller molecules and to absorb smaller molecules to facilitate absorption. What is the purpose of having holes in some septae? Enables movement of mitochondria, ribosomes, and nucleic acids. What do you call fungi lack septae? Cenocytic fungi. What is the purpose of having hostoria in some fungi? Purpose of having hostoria to penetrate and absorb or exchange nutrients between plants and the fungi. To penetrate and absorb an exchange of nutrients between plants and fungi. What is a fungal mycelia? Network of branch hyphae. Network of branch hyphae adapted for absorption of nutrients. Right, next one, part C1. Following dichotomous key prepared by a student to study features of fungi for Allomyces, Rhizopus, Saccharomyces, Aspergillus, and Agaricus. Allomyces is Chytridiomycota, Rhizopus, Zygomycota, Saccharomyces, Ascomycota, Aspergillus, Ascomycota, Agaricus, Basidiomycota. Unicellular fungus, what do you say unicellular? When you say unicellular, Saccharomyces is unicellular. Allomyces is multicellular, you can't take that. Multicellular fungi, rest of the one. So it goes to number 2. Number 2, no septae kyanne, then you have 2, Allomyces and Rhizopus both. No septae, so it goes to number 3. Then septae present means the remaining one. So you remove Saccharomyces. Now from the remaining, Allomyces and Rhizopus goes to number 3. Septae present, the Aspergillus and Agaricus. Zoospores are produced, that is Allomyces. Zoospores are not produced by Rhizopus. Then asexual reproduction is present, Aspergillus. Asexual reproduction not common, Agaricus. So this is how the marks allocated for that part. Right, question 2. What is the difference in nutrition of Chytridiomycota and Zygomycota? What is the difference in the nutrition of chytridiomycota and zygomycota? Difference in the nutrition of chytridiomycota and zygomycota. Chytridiomycota, some are decomposers, others are parasites. Difference between chytridiomycota and zygomycota. What is the difference in nutrition? So this is correct. Some are decomposers and others are parasite. Zygomycota, most of them are saprophytes. Some are parasitic or show commensalism. Next one, what are sporangiophores? Sporangiophores. Sporangiospores. This is now you can see people in ascomycota. Sporangiophores. Sporangiophore. Sporangiophore neda. Araka conidiospores. So I'm wrong there. Sporangiophores. Let's read that part a little bit, people. Sporangiophores, zygomycota, asexual reproduction. They produce sporangiophores, endogenous, asexual spores. I'll read that part. Zygomycota, asexual reproduction produce sporangia in which genetically identical haploid spores are endogenously produced called sporangiophores. So genetically identical haploid spores endogenously produced in zygomycota. So 1 point for that. State special characters of zygosporangia. Special characters of zygosporangia. Resistant to unfavorable environmental condition. Multinucleated structure. Metabolically inactive. Study structure produced by plasma. Sturdy neither, not study. Sturdy, please correct that, people. This is sturdy structure. STURDY. Sturdy structure. Structure produced by plasmogamy and karyogamy. State two differences of spores produced by ascomycota and basidiomycota. Differences between ascomycota and basidiomycota. Ascomycota produce inside the ascus. 8 ascospores are produced. Basidiomycota produce exogenously on the basidium. Neither. 4 spores are going to produce. What is basidiocarp? Basidiocarp is the fruiting body produced during asexual reproduction of basidiomycota. Where basidia form in basidiocarp? On the gills. Where basidia are formed in basidiocarps? On the gills. Question number 2A. What are most important colors in regulating photomorphogenesis? Red and blue light. 2 marks for that. What are photoreceptors important for photomorphogenesis? Photoreceptors, cytochrome and phytochromes. Or else you can write blue light photoreceptors and red light photoreceptors. State the role of photoreceptors in seed germination. Seed germination, you can mention several roles, neither. Now, one is the red light photoreceptors are involving in the seed germination, especially 660 wavelength light, initiate the seed germination process. Far red light is going to inhibit the seed germination. Hypocotyle elongation is inhibited by the blue light photoreceptors. So those things you have to write here. Because of forest canopy Why plants grow under shade receives less red light. Reason is forest canopy absorb red light for photosynthesis. So what comes down is the far red light. Neither. What photoreceptors involving in phototropism? Phototropism, blue light photoreceptors. What are statolith? Specialized plastids containing dense starch grains. Right, next one, what is the role of calcium in lateral transport of oxygen? Now, you know, people, in the growth of the root, calcium go and accumulate on the side, right? And then decreases the growth on that side, neither. Redistribution under effect of the statolith aggregation. Right, calcium, once it accumulate on the direction where the statolith aggregate, so this is now resulting people the auxin accumulation on the same side, suppressing the root growth, right? So this is helping in the root to bend towards the gravity. So this calcium lateral transportation of auxin, redistribution under the effect of statolith aggregation, calcium redistribute, right? So I think that statement given in the resource, gravitropism, right? Gravitropism, it's mentioned. Calcium causes lateral transport of auxin with the root as aggregation of statolith, the lower point of the root cap cells triggers redistribution of calcium ions. Part B, people state the plant hormone involving in following functions. Regulate development of fruits, auxin, stimulate fruit growth, gibberellin, stimulate seed development and germination, gibberellin, stimulate seed germination, cytokinin, development and germination, gibberellin, germination only cytokinin, retard abscission, people, auxin, promote leaf abscission, ethylene, delay leaf abscission, gibberellin, promote leaf senescence, abscisic acid. What is triple response in seedling? Inhibition of the stem elongation, promote lateral expansion and horizontal growth. What are hormones? Plant hormones, neither. Signaling molecules produced in small quantities, get transported from the place where they are produced and trigger responses. So that 4 marks for those highlighted underlined parts. State plant responses to drought stress. Plant responses to drought stress. Plant response to drought stress. Right, under the water deficit condition, synthesis and release of the abscisic acid, this act on the guard cells and close stoma to reduce transpiration. In grasses, right, this is one response. In grasses, leaves roll. Some plants shed their leaves in seasonal drought. State the differences in plant responses to freezing and cold. Freezing, increase the cytoplasmic level of specific solutes, which help to reduce the water loss. Then specific proportion of, increased proportion of unsaturated fatty acid to keep the membrane more fluidic at low temperature. State how halophytes tolerate salt. How halophytes tolerate salt. One is develop salt glands to secrete excess salt out, right? That's only one, neither. Salt stress, but then the halophytes, they have the salt glands. What is the common chemical formed by plants in pre-existing mechanism? Phenolic compounds, neither. Pre-existing mechanism and induced mechanism, both phenolic compounds. What changes occur in the cell wall in induced mechanism? What changes occur in the cell wall in induced mechanism? Induce morphological changes take place. Induce. Now, when the attack continue, what changes occur? They say that the morphological changes in the cell wall given straight away. So that's the answer. Right. Then people, we have to go to essay questions. Essay questions, describe C4 photosynthesis. So when you describe C4 photosynthesis, you have to describe the special pathway present between the bundle sheath cells and the mesophyll cell. Plus people, what happened inside the bundle sheath cell, you have to describe too. C4 plants, photosynthesis involve initial carbon dioxide fixation in mesophyll cells using PEP carboxylase and as carbon dioxide receptor. This result oxaloacetic acid, which forms the basis for the metabolic pathway. Oxaloacetic acid, it is a 4-carbon compound and therefore you say C4 pathway. Oxaloacetic acid is rapidly converted to malate that diffuse into bundle sheath. Here the carbon dioxide released by decarboxylating enzyme to form the pyruvate, and then this carbon dioxide is fixed by Rubisco in the bundle sheath cell chloroplast, and then malate after releasing carbon dioxide become pyruvate. Pyruvate diffuses back into the leaf mesophyll cells and receive phosphate from ATP and then regenerate PEP. C4 plants exhibit special Kranz anatomy where mesophyll cells and the bundle sheath cells are tightly interconnected. Bundle sheath cells enclose vascular bundles and surrounded by mesophyll cells. The bundle sheath cells have expanded physiological function. The higher organelle content is there. The chloroplasts in the mesophyll cells are different from bundle sheath cell chloroplast. The chloroplasts of the mesophyll cells are different from bundle sheath chloroplast. Mesophyll cell chloroplasts carry out only light reaction, and bundle sheath chloroplast possess So it generally says describe C4 photosynthesis, right? So you can write the significances. Bundle sheath chloroplast possess very few less differentiated grana containing only PS1 but not PS2. This pathway helps plant to improve carbon dioxide fixation by preventing photorespiration in hot dry climates. The PEP carboxylase enzyme is much more efficient for carbon dioxide fixation and has no affinity for oxygen because Rubisco operate at high carbon dioxide concentration in the bundle sheath cells. It works more efficiently than C3 plants. Consequently, less enzyme provide better nitrogen use efficiency. People here the both C3 Rubisco, what happened inside the what you call Kelvin cycle that happens in the bundle sheath must be described too. Let me check this again, people. Describe C4 photosynthesis. Right. So what we have to mention, pyruvate diffuse back into mesophyll cells. C4 plants exhibit right now. Before this point, I think we must mention the carbon dioxide then enters into the mesophills bundle sheath chloroplast enters into bundle sheath. Refix kila method tino neda. Refix by Rubisco, which exclusive hari. Okay, it's given. Right. So both C3, what do you call the Kelvin cycle, need to be described, right? You don't have to write it in detail, right? But it must be included in the answer. Now we are moving people to the next essay question. Next essay question. Second question, describe the plant responses to light and gravity. So this is like summarized. Now there are a lot of plant responses. We don't take them all. Now here only two. Plant responses to light and gravity. That's all. So light, we discuss people plant responses to red light and blue light. Right? You have to mention. So photomorphogenesis is the key event of growth and development of plants triggered by light. Action spectrum reveals that red and blue light are more important. And then in photomorphogenesis occur with two major classes photoreceptors, namely cryptochrome and the phytochrome. Cryptochrome are blue light photoreceptors. Phytochromes are red light photoreceptors. Phytochromes are major type. And then they are controlling seed germination, shade avoidance, and flowering. Light-induced slowing down of hypocortical elongation is done by blue light photoreceptors. As nutrient reserves are limited, many types of seeds germinate when only light is optimum. So red light 660 increases the germination, 730 inhibit germination. This is due to the effect of phytochromes. During seed germination, when the seed break the ground, light-induced slowing of the hypocortical elongation due to blue light photoreceptors. So the seed germination, it's affected by two ways. We discussed all these things last week discussion. Flowing of the field and death of the shady tree can promote the seeds germination. All seeds don't need light for germination, some seeds only. Phytochromes provide the plant with the information about quality of the light, which enables the plant to get adapted to changes in outside light condition. Shade avoidance response of a forest tree that requires relatively high light intensity. Forest canopy absorb more light, allowing only far red light to pass through. So therefore, the tree below the canopy allocate more resources to grow taller. So this is plant spacing and to increase the plant height. Photoperiodism is the intervals of 24-hour period in which the plants are exposed to light, affecting in people flowering of many plants. Controlled by phytochromes. Right? The growth of the shoot towards the light is positive, away from the light is negative. Positive photorealism strengthens photosynthesis, and this response result in differential growth of the cells in the opposite side of the shoot. So when light comes from one direction, opposite side growth is going to enhance. Darker side elongates faster than the brighter side. Blue light photoreceptors involved in phototropism, blue light photoreceptors involved to bend the shoot towards the light. Gravitropism can be either positive or negative. Root display positive gravitropism, shoots display negative gravitropism. So gravitropism occurs with the seed germination. Plants detect gravity by settling statolith. They can settle under the gravity at the lower portion of the cell. In roots, the shoot takata, they have not described any mechanism. But in roots, the aggregation of statolith on the lower point triggers redistribution of calcium ions, which cause lateral transport of auxin, and as a result, calcium and auxin get accumulated on the lower side, and lower side elongation now becomes slower. Neither. As a result, root is now bent towards the gravity. At high concentration of auxin, cell elongation inhibited. As a result, a slow growth, and consequently root grow towards the gravity or downwards. Right? So that's all people for the day. So this is paper 10. So we have discussed 10 papers. True that people, it's not full papers. It doesn't matter, right? They are not full papers, it doesn't matter. We are doing papers now. It's extra work to the normal classwork we do at the moment, right? And we discuss every time people, when you do questions only, you find difficulties and you find the places where actually where you cannot remember, where you have not studied enough, where you have not learned properly. Those things you can find only by doing papers, right? Papers are not to do at the last few months. That is not going to work, right? Now itself, you have to start it slowly and then do it slowly. Then the exams are going to be people quite easy. It's like a small chess game. You don't have to prepare separately for exams if you do this properly with me. So with your support, if you support, we can make this a success. Your support is very important. I can't teach here alone when no one is there, right? There must be some people to listen and then to participate in this work. So 10 paper. So we can do every week if you do a paper. People, you have how many months for the exam? How many months for exam now, people? 10 months, 10 months, right? I put a countdown. So countdown is not 1 year now because the exam is on August. So it's going to be 10 months. So this is high time, right? It's not too early. This is high time, right? Otherwise, you are never going to be ready to do papers, right? So keep that in mind. Support this process. I'm with you every time to help you, right? So we'll do this continuously. Thank you very much for your participation. I'm going to meet you soon, right? I'll meet you soon on Saturday, right? Okay then. Thank you and bye-bye.